Hi all, welcome to this week's asynchronous lesson on CRISPR-Cas9. This will complement last week's click and learn activity very well. CRISPR-Cas9, commonly referred to as just CRISPR, is often mentioned in the news for its potential to treat genetic diseases. In this activity, you will explore and learn about this biotechnology tool by building a two-dimensional paper model of the CRISPR system. You will then apply your knowledge of complementary base pairing to model how CRISPR targets specific DNA sequences. Last week, we learned that the CRISPR-Cas9 system can be used to target specific genes in an organism's genome. CRISPR-9 can be designed to either inactivate or knock out genes or to edit genes. CRISPR-Cas9 is a versatile biotechnology tool that can be easily modified to meet the needs of a research project. CRISPR-Cas9 is a technology that allows scientists to edit a cell's DNA. Since it was first described in 2012, CRISPR has generated much interest for both its exciting potential for treating genetic diseases as well as for the potential ethical and safety concerns such as creating designer babies and superhumans. The CRISPR system was first discovered in bacteria, where it functions as a type of immune system. Scientists have modified the bacterial system to produce a biotechnology tool for editing DNA. CRISPR-based technologies are widely used in research and have been applied in a broad range of biological studies. Treatments using CRISPR technology are also being developed for several genetic diseases, including sickle cell disease and cystic fibrosis. The technology is relatively cheap, easy to use, and allows researchers to ask new questions and get results faster than previously possible. The paper model in this activity shows some of the key components and processes in the CRISPR system. The activity explores two different applications of CRISPR: gene inactivation knockout and gene editing, and how they make use of the cell's processes for repairing double-stranded DNA breaks. Gene inactivation with CRISPR makes use of non-homologous end joining, or NHEJ, which is the cell's main repair process for fixing double-stranded DNA breaks. You may wonder how CRISPR can cause mutations using this process. During NHEJ, the broken ends of DNA are brought together and rejoined, but this process can be error-prone because sometimes nucleotides are lost from the broken ends and are re-added incorrectly by the cell's repair machinery. If the DNA sequence is repaired correctly by NHEJ, Cas9 will just bind to the sequence using the guide RNA and cut the DNA again. Although the cell can keep repairing the DNA, Cas9 will continue cutting it until the cell finally adds the wrong nucleotides, often causing the gene to lose function. Once the DNA sequence has the wrong nucleotides, Cas9 will no longer cut it again because the guide RNA will no longer match and bind to the DNA. Gene editing with CRISPR makes use of a homology-directed repair, or HDR, a repair process that cells typically use only shortly after DNA replication in the S and G2 phases when sister chromatids are present and can serve as templates for repairing the breaks. Since HDR repairs breaks by copying from a homologous template, it is more accurate than NHEJ. When using the CRISPR-Cas9 technology, scientists supply the homologous template in the form of donor DNA so that the cell will use HDR rather than NHEJ to repair the double-stranded DNA break. Scientists can design the homologous template to include a new sequence or correct an existing sequence in a cell. This week's activity shows how two specific DNA sequences could be targeted using CRISPR-Cas9 technology. Both of these target sequences are from real genes. The target DNA1 sequence is from the human MC1R gene. This gene is associated with melanin production in skin and hair. In this activity, CRISPR-Cas9 is used to inactivate the gene, which would be expected to cause a change in coloration. The target DNA 2 sequence is from a gene called MYBPC3. A deletion of 4 bases, GAGT, in this gene causes a genetic disease called hypertrophic cardiomyopathy. This disease occurs in 1 out of every 500 births, making it one of the most common genetic heart conditions. It can result in heart attacks in adults and is a common cause of sudden death in otherwise healthy young athletes. As modeled in this activity, CRISPR-Cas9 has been used to edit the mutant MYBPC3 gene in human embryonic cells by inserting the missing GAGT sequence. Note that the paper model makes certain simplifications in its representation of CRISPR, as you will probably notice if you compare this to the click and learn activity from last week. For example, actual guide RNAs are much longer than what is shown in the paper model. In reality, each guide RNA forms a complex three-dimensional shape containing several double-stranded loops and twists. This three-dimensional shape allows the guide RNA to bind to the nuclease Cas9, similar to how a substrate binds to an enzyme. In the cell, Cas9 binds to PAM sequences, which occur throughout the genome. When it binds PAM, Cas9 unwinds the double-stranded DNA. This step is not shown in the paper model. If the guide RNA matches the target DNA, it will bind to one of the 2 DNA strands by complementary base pairing to form a DNA-RNA helix. This step is also not shown in the model. And finally, DNA repair pathways are more complex than they appear in this model. In the part of the model involving HDR, for example, it may look as if the donor DNA is spliced directly into the target DNA and replaces parts of the target sequence. In reality, though, the repair involves homologous recombination between the donor and target DNA. Now you're ready to start your modeling. Good luck!